So hi, hello and welcome again. Microbehunter here and welcome to another Saturday live stream. Well, this live stream today is going to be a little bit different because it's almost live, so to say, because I've pre-recorded it. But you are free, of course, to participate in the live chat. I would like to send a hello again all around the world. And a little bit of background info why I'm changing the format around just for today. Again, a little bit is because for time reason I don't have time during the live stream time itself, so I decided to pre-record it just to keep the pacing and the rhythm of the live stream the same every week. So I've pre-recorded this live stream, so if you ask any questions and type any questions into the chat, then I'm sure there will be many other members in the community who are willing to answer. And then later on, if you have any questions that you want me to answer, well, I'm not able to answer live in the chat. There is, of course, the possibility for me to pick up on those questions and then later make a separate episode, and then I can answer your questions. So please feel free in any case. What am I going to show you today? I'm going to do a little bit of staining today again, and I've got over here in the background 6 different stains, and over here, these are basically the stems of a plant. And what I would like to do is I would like to experiment now with a new microtome principle. You probably know those microtomes here. This is a homemade one, but I'm not going to be using those. I'm going to be using this this laundry clip as a microtome, and I would like to see how this actually works. I think it does prove to be promising, and it's a very simple thing to do. So for that reason, I would like to share this with you a little bit, and then you can improve on it yet further. So that's basically what I would like to do today, and then also maybe towards the end of this live stream, or this almost live stream rather, I would like to start to grow again the so-called the slime molds. Because I've got those slime molds here in dry on a piece of filter paper. You can actually see them over here. I would like to see if I'm able to get them back to life again. So I'm just going to start to rehydrate them, and then maybe in a few days I can show you in a different video on how well that went. But I would like to share this also with you today. So let's get started, I would say. So what did I do here? Well, I collected a plant. It was called the so-called sword lily. Not so relevant now. It's a monocot plant, and what I did, I cut the stem into different parts and put it into alcohol, and it started to take out the chlorophyll and of course also the water. So this means that's why it's a little bit greenish. What I've done is over the last couple of days I have simply depigmented it, and I've always after every day I've exchanged. Be careful that it doesn't run out. I've always exchanged the alcohol, and you can see that it has turned quite green, and then I also always refilled it again with fresh alcohol. And this way it started to not only lose water but also the pigment, and that's fine. What I would like to do now is I would like to make a cross section of this stem here, and I would like to first show you how I've done this with this homemade microtome here. And there is actually a little bit of a disadvantage here that I would also like to show you, then I'm going to show you this clip method. And the first thing that we have to try is, does this even fit in here? You see, it's too tight. And so what I'm going to do is I'm just going to take a smaller one over here, and let's put the other one back, and lid goes on top because I do not want anything to run out. And all you do is you basically lower this here. You put the stem in here, and this is actually the method that does not quite work as well. And what you now need is you need an absolutely fresh razor blade, and that's of course the dangerous part because they're sharp on both sides. So maybe it would be a good idea to maybe cover one edge over here with maybe some tape, some sticky tape. Let me do that right now, actually, as a matter of fact. I mean, those razor blades are way better than, for example, so-called cutting board knives, because they're way sharper, with all of the consequences, of course. And I just flip it around here. so that when I'm holding the razor blade here like this, that I don't accidentally cut myself, right? And this is now where things start to become a little bit difficult, because what you have to do is you have to now make a cut here. And I found that essentially it's difficult to do because the outside of the stem is fairly hard, and the alcohol also makes it yet harder. It dehydrates the stem, and for this reason, it's pretty difficult to actually cut across, I found. Yeah? So this is actually, especially maybe the first one over here, but it does work eventually. You probably want to save this here, okay? So I'm going to put that back as well. You know what I'm going to do? Yeah, I'm just going to drop it back in here. Yeah. And then what you need to do next is you need to advance it up a little bit here. And you can actually see that it's actually already lower than the surface, than the washer here. And I think that has to do because as it starts to dry up, the alcohol, it starts to shrink. And then we have again the problem that it becomes yet harder. We have to maybe... And again, this is not going to replace a proper, let me say, a proper microtome, okay? Yeah, but you can actually make slices like this, which are... Okay, now I dropped on here in the paper. You can make slices like this, which are actually not quite as thin, but maybe thin enough for our demonstration purposes. Okay, so I'll be simply doing this, and as a matter of fact, it does kind of work. And what you want to do next is you don't want to just let it dry, but you want to put it actually on a dish with a little bit of water as well to rehydrate it, and also to remove the alcohol. Okay, now I just realized that there is not enough water in here. Here we go. Okay, that'll do. So, and that's basically all we do is we just take it and we put it in here. And this other one here as well. Up. Oh, I dropped it. Here it is. Can I pick it up? Do you actually see how it's moving around? That's because of the surface tension of the alcohol mixing with water, right? So that's basically the one possibility here. But you see, it doesn't really quite cut straight. You know, sometimes when you cut, you're kind of lifting it up a little bit, and it doesn't have to be completely perfect either. I just want to be able to see a few cells. So maybe sometimes cutting it a little bit. Here it is now. Diagonally is also fine. But the disadvantage of this method, and that's why I'm not going to be using it, is because when you take it out here, you're actually going to see that it starts to push in the back side here. That's the screw that we have in here, right? Starts to push in the bottom part of the stem. So that's not really consistent, right? So for this reason, I want to show you now a different method, which I think works also quite surprisingly well. I'm just going to take a new stem here. Okay. I don't know, maybe this one over here, because it has also just been rehydrated. Not rehydrated, but actually taken up some alcohol again. And then look what I'm doing next. I'm just going to take this clip here. Let me put this here away. And you're just going to clip it in here. And that's going to be our new, here we go. I have to move the camera over a little bit. Maybe let me just see if I can, So, that's basically all you do is you just clamp it in. It's going to be squeezed a little bit because of the pressure, but I think even that is okay. And then what you do is you just, do the same thing. You take the razor blade. And and I found that this actually works. It works better. This is still too thick, and then you have to kind of carefully push it out a little bit, and then it's kind of trial and error, because some of them are going to be thicker, some of them are going to be thinner, but they'll be fine. You just basically then pick those that work best. Yeah. I mean, and it doesn't have to be perfect either, but I think it works. Works fine. Just put it in here. And as a matter of fact, I actually got results that were surprisingly good. So I have to make what, at least 6, 7, or 10 of them. The reason being that, maybe some of them are going to be too thick. Ooh, okay, that's 2 of them now. So what I've basically been doing is you just basically push it out a little bit. And of course, it does not. This is not a technique to use for for the production of large amounts of these cross sections. I'm just for your info, I'm always looking down. That's why I know you're supposed to keep eye contact with the viewer by looking up into the camera. I can also try to do this and cut this by looking at the screen. It actually also works. Yeah. But if the stem does become harder and harder and harder, this is a fairly thick one. It's way too thick. Yeah. So, and a little bit here again. So let's give it another try. So you see, maybe cutting it a little bit, just a little wedge or so is also going to be fine. It's not necessary to have the complete cross section. It's all I want to have is a couple of cells, because I won't be able to look at the whole thing anyway. And and trial and error means that you're going statistics. If you made maybe 20 cuts like this, maybe 5 or 6 of them are going to be fine. But what is important is it's reasonably thin, and you of course you can try to cut this without any tools as well, without a microtome. But I think you really end up cutting yourself a little bit. So yeah, I mean, this kind of. Let's make a few more. Oh yeah, here it is. Here it is. So let's do another one here. I'm not going to edit out all of these things here, because I do want to kind of keep a little bit live stream vibe. And by taking my time cutting them, it also gives you time, of course, to write comments or suggestions. Oh, there's something sticking over here. 1, 2, 3, 4. Maybe there'll be enough already. But you also see that you don't really need a lot. And let's have a look here. Let's cause another wedge. Okay. And even if it's not perfect, it doesn't matter. I just want to show you a little bit with these live streams, or in this case, these premiere videos. I just want to show you kind of a proof of concept, and then to motivate you. And then you can pick it up from there and then improve on that. For example, one of the things that one might do, I did not have the time yet to do this, but I've got a washer here. It does have some edges that you might want to sand away, but maybe using some epoxy glue, it would be possible maybe to glue it like this, so that one edge just aligns with the clamp, and then just glue it on one side here, right? And then you have something, you're able to do it like this, So, and I think it's important that it's not centered, right? Because if it is centered, then again, there's a stability problem, but that it actually aligns with the one side of the clamp. So then if you have it like this, then you're able to cut it like this, and then kind of the washer, so acts as a support for the stem. So that's something I've been thinking about as well. Didn't do it yet, but So what we can try as well is something as follows. I'm going to put this again back here. So you see how much, it's already shrunk quite a bit, not only of the pressure, but also because of the evaporating alcohol. Doesn't matter, when you put it back, it will, and I will have to refill it again with fresh alcohol. But yesterday or the day before yesterday, look what I've done. I've taken, one of them, and I, after it was in alcohol, and I put it in here into water. Now that's of course a risky thing, because if you've got water, then you of course also have the growth of microorganisms. But let's give this here a try. So let's see if how well this actually works. And I think it does cut much more easily. Because actually way better. Let me see. Let's see how well this actually works. So basically, I would suggest probably also try to rehydrate it, because before you actually cut it, because this also changes the consistency quite a bit. Just look at this. It's actually way thinner. Yeah. So this might actually also be a viable solution, so to say. Again here. Okay. So what I'll be doing now in this is actually way. Actually, it does work. Yeah, we'll have to see how well it works. But this does seem to be a possibility. Let's make a few more. The reason why I like to make those plant cross sections is because they look indeed quite pretty. Okay, yeah. Let me try again to make a few cuts. Refocus this. So I'm gonna try something different now because I'm always looking down on my table. But now let's have a look and let's look up a little bit here. I'm trying to cut it now, looking at my computer screen. That's actually quite good. So the suggestion that I have, therefore, is maybe keep it in alcohol a little bit to take away the pigments, and also for long-term storage. But maybe for cutting it, it would be better to quickly before it rehydrated. But always be aware when you add water, then you of course also have bacteria starting to grow and decomposing it, and then it's going to turn bad. Okay, I think we're just going. We've got enough. I think you get the idea. Okay, and let's put them in here. And yeah, plenty of stuff floating around here. But what's this one on here on the side is? That's too thick. We're just gonna throw that away. Okay, yeah, and we're gonna take it from here. Okay, so this is basically the new how do you say suggestion on how you can grow, not grow, how you can cut your yeah. If you actually look at this here, it's not quite as clear anymore, right? It's a little bit cloudy. This actually means that there's some bacteria already started to grow in here. Yeah, so I probably want to dump it out again. So let's do some staining now. Okay. So for staining, I'll be using the following technique. Okay. I will be adding the stain to those, or I'll be adding those things to the stain, and then I'm going to give it a few minutes, and then I have to wash it. Washing it basically means I'm going to rinse it in water. The reason being that otherwise there's going to be too much stain, obviously, right? So this is the thing that I'll be doing, and for this reason I need also some kind of a wash water solution, which I have got over here. I just realized I probably need different ones for the different stains, but I think they'll be okay. Yeah. So let's get started. I need a slide, so I'll be doing the staining directly on a slide, but then I will be putting it on a different slide. So, and I don't know if I'm able to remember all of these things that I'll be doing, so I'll be placing one here. I'll be placing another one over here. Okay, I don't know, maybe I should actually put all of them. Let's do a quality. Why not do a quality check first, whether it's actually thin enough. And again, this is a little bit of a proof of concept. Okay, so maybe it'll work, maybe it won't quite work as nicely. So I have to choose now fairly thin cross sections. You see, or I'm over here. So which ones am I going to choose? Maybe this one over here. So, okay, let's do the following. Let's have a look at them. Yeah, this might be a little bit thick still. So let's have a quick look at them under the microscope for first quality check. So let's see where it is. So here we go. I need to switch on the microscope. Here we go. The arrow I don't want to have. So let's have a look. It's now at low power. I think low power is going to be fine. This is a nice one. Look, in the center, the white area I've cut away too much, so the center was lost, but that's okay. Okay, let me remove the filter a little bit. So that's number one. Number two, it's already a little bit thicker. But the cells over here on the side will be fine. I just want the purpose of this experiment or live stream is simply to test which type of stain actually works best. And that's actually something that I've also not done in that systematic way before. Lots of air bubbles here. That's interesting. Were there no air bubbles in the other one? Could this have done to do something a little bit with whether it was an alcohol or not? That's actually something new that I also have to figure out a little bit. And so there are air bubbles here. It's something that I don't know. Does alcohol remove the air bubbles or not? This would actually be a very nice specimen, but there are lots of bubbles here. And here as well. So, but let's just go in a little bit and have a look. Yeah, actually, the cells themselves are fairly well cut. So I'm actually on a cellular level. I can imagine many of the cells are also running vertically, especially the xylem. So of course you're going to get the nice cross sections this way. Yeah, but yeah, that's basically how it looks. Yeah, and we're now going to be adding the stain, and I think I might have to label it somehow. Where did I put my marker? Here it is. Okay, so but let's do this one by one, maybe. Otherwise, it's going to be too much. So the first one is going to be the Pelikan ink. This is just regular fountain pen ink. Okay, so I'm going to be adding a drop here. And the next one is Löffler's solution, which is basically methylene blue plus alcohol. Plus potassium hydroxide. There is a specific formulation. And here I'm also going to give it a little bit of time. And what I'm going to do now is I'm going to label this here. So the first one was ink, just regular ink. And this one over here was Löffler's methylene. Methylene blue. I don't know. Yeah, it's Löffler. Löffler. Or maybe just an L would have been enough. Yeah? So that's basically, yeah, we're just going to give it a minute or so. And I'm going to take it one by one. I would say, otherwise there's going to be too much chaos here. And then I need to just say that maybe it's been enough. I found it. Here it is. Okay. And now I'm going to just rinse it. Yeah, and put it on here. Okay. I'm going to do the same thing over here with this one here as well. And we're going to put it in here, and then we're going to make a decision which one is going to actually work best. Because I might be going up a little bit with the magnification also higher, I need to put a cover glass on here. Just for first try under the microscope, it'll be fine without cover glass. But cover glass is always a good idea, because otherwise there's also the danger that you're going to make the objective wet, something you don't want. And another thing that I would like to do is I would like to add a little bit more water here. So I need my pipette, and I need a little bit of water. So I need to, great. I forgot to just do the following. Okay, I'm gonna use here. This is going to be that's my water, and I'll be adding a little bit of water beneath the cover glass. And here as well. So let's give it a try, okay? Let's see how nice or not so nice it looks. Where is my? I have to switch over again to the scope view. Yeah, so I suggest generally experiment around. You use just fountain pen ink as well, different types of stains and inks, and that's basically how it looks like. And I can already tell you it's not the way that I want it to look like. I mean, there is because because the cell walls did not really take up the color very well. That's why we're experimenting around. Okay. So, just for those of you who are a little bit interested a little bit in biology as well, because sometimes it's a little bit more fun if you know a little bit of what actually is going on. So I'm going to switch on the arrow, but don't worry, not too much biology. These things in here, of course, are the individual cells. They have a cell wall. That's why they're quite easily visible. And those things that you see over here are called the vascular bundles. This is basically where you have cells, long cells, that carry up water up the plant, and also nutrients like the sugar that was made by the leaves down the plant. So the two types of vessels are called xylem and phloem. Xylem transporting the water up, and phloem transporting it down. But what we see over here with this staining, with using the ink, I mean, it does look a little bit blue in the background, but actually the actual cell walls themselves, they did not really take on the ink very well. Let's just go up here a little bit. So I don't know. We're going to see, and we're going to compare it now with the others. By the way, I do not have any filters right now. I'm just in regular bright field. Yeah. So this is now. Let's try it with a Löffler's solution. And here, that's as expected. That's methylene blue. We can already see. I mean, we have to look at the bubbles, right? I mean, the bubbles also look kind of, I don't know, nice. But here we can already see some two different interesting things that happened. Number one, some of the cell walls actually are more blue, but more notably, sometimes also the cell's contents has been stained. So look. Sometimes the inside of the cell is white. That's basically, and sometimes it's also blue, because the cell membrane was still intact. And you can also see that the cell wall is a little bit more blue. So generally I feel, here again the vascular bundle. You can also see that when focusing there is a certain thickness to the cells as well. So we see that take-home message, methylene blue works better than fountain pen ink. However, I do have to tell you that fountain pen ink does also have its merits. It can be used, for example, to stain cheek cells quite well, but evidently not plant cells. So that's the methylene blue, and quite, I would say, quite nice looking again here. Those vascular bundles. And let's compare it again with the fountain pen. That's the fountain pen ink. You see, it did not properly react. I don't know. Maybe the background, some of it was a little bit maybe stained. I don't know. So, and again here, that's the methylene blue. Here, these are the bubbles. There are different ways of trying to remove the bubbles. So what I've tried is also in a vacuum, for example, maybe taking a syringe. For example, I've got a syringe over here. And then trying to put it in there, and then holding it, and then pulling on the syringe to create a vacuum or a small vacuum chamber. So there are many hobbyists that have experimented also with that. So, but let's move on a little bit. I still want to do a little bit more staining. Okay, and what I see here is I'm going to put this away, and let's. I'm gonna put this away here, and then let's go on to the next one. I have to put it somewhere where I'm not. So the next one would be iodine and acetocarmine. Acetocarmine actually is not is actually used to stain chromosomes, which we're not going to see here. But this one is iodine, which is actually used for staining starch. So we're probably not going to see a lot here. So my expectations are fairly low for this. And this one over here is actually used for, as I mentioned, for staining chromosomes and DNA, not intended for these cells here. But we're going to still give it a try. And we kind of hope a little bit maybe to, if there has been, especially for the iodine one, if there has been photosynthesis happening in the stem, then what we're also able to, then we expect starch to be deposited there. And when there's starch there, then these starch grains or this starch should actually stain black or dark blue or black. And we're going to then have a look. And as a matter of fact, if you already see that there is on the outside, as expected, I will explain. You see that it's already starting to become a little bit darker here. When working with iodine, always be a little bit careful because iodine is quite corrosive. And for this reason, you really want to make sure that it's properly washed so that it doesn't damage any of your microscope parts if accidentally it comes in contact with those. So I'm going to now do the following. I'm going to again put those stains away, simply to clear. And this washing solution is still kind of okay, so I guess it's gonna be fine. And I don't know, should I actually touch it with the tweezers here? Because I do not want it to get in contact with metal. So I'm going to use first some disposable. Maybe I'm overdoing this a bit. I'm going to just lift it out and put it into the water over here. So then it's been kind of washed and rinsed. All of the excess iodine has been removed. We see already there was some kind of a nice staining reaction happening here. Here we go. Acetocarmine also contains a lot of acetate, but I think that's going to be fine. Just quickly take it up and put it in here. It's difficult to see. Now, if you don't rinse it and if you don't destain it, then there is, of course, the possibility that everything is going to be so pigmented and stained that you're not able to see anything at all. So we have to do the following: iodine and acetocarmine, AC. Okay, again, a cover glass goes on top. Again, if you feel free to comment in the chat. Maybe I'm going to be answering later on, but I need to get the water again. Where's the water? Here it is. Here it is, and here it is. Okay, here we go. So let's put it again under the microscope and let's have a look. This is now the scope view. Every time when I switch around, I don't know where to find it. So here we go. So that's again the Löffler solution, the methylene blue. See that? And now let's give a try for the other one. I'm switching down again with the So this here is now the iodine. Okay, let's have a look. Actually, here we go. Let's go up. We have to be a little bit, honestly. So we have to ignore the bubbles again. So pretty much everything's yellow. But what about here on the side? I need to now really turn up the brightness. I mean, you do see that the cell walls here on the outside do seem to stain darker, but something what I kind of was hoping to see, I'm not seeing. I was hoping to see some kind of starch deposits. Why? Because the outside was green, and but maybe I do see them. Let me go up a little bit with the magnification again, and it's not very Could it be because on the outside it's green and therefore photosynthesis is happening there? I don't know if these in here on the outside are actually tiny little starch grains. If you cut, of course, the stem to thin, so that the cells are cut open, then of course also the contents are lost. I have to go open the condenser yet more. I don't know if this is this actually looks a little bit more, for example, something like this here, right? This actually looks, you see it on the outside you have the cell wall, right? And on the inside you have this other ring here. Sometimes, especially after you've treated it with alcohol, you see that the cell's contents starts to shrink. And then if this happens, then the cell membrane separates from the cell wall. So this maybe is the case over here, and maybe it could be that maybe those dark dots on the outside here, which you actually see mostly on the cells here on the periphery, not so much on the cells here further in the center. These dark dots, which I hope or assume could be starch grains. Okay, because the outside is green because this is where photosynthesis happens. You actually see it over here. Yeah. But again, of course, it's fairly thick, the specimen, but if I were to have cut it thinner, then of course this would have also resulted in the cells being cut open and then with a loss of cells contents. Yeah. So if you see that there is some stuff in there, right, in those cells here, but not in those that are further in the center here. Yeah. So let me just go around the periphery here. Maybe I'm able to find a so that is a short reminder. That is, again, here we see it a little bit better. That's iodine. Okay. Iodine staining, which is selective for starch. Yeah. So maybe it's easier or better if I were to do this for leaves. Yeah. So, but we now have acetocarmine. Yeah. So that's again the other one. So acetocarmine, which is actually used for staining DNA. So my expectations are really not high here. My expectations are not high here, and it almost looks like if it's not stained. So why is this still of relevancy? Because it shows that you choose, of course, the stain based on the substance or the components or the parts that you want to make better visible. Okay. But there is, look, that's interesting as well. You see that those cells here, they seem to have a different color now. Yeah. But that's not because of the chlorophyll so much, because that would be actually not in the cell wall. The chlorophyll actually is in the chloroplasts on the inside. So I wonder why this ring over here looks slightly yellowish greenish. Yeah. So that is basically our acetocarmine, and as we can see, not a lot of staining has happened here. So let's move on to the last two stains. I need to go again back to the desk view. So again, I have to put the slides somewhere where, So, and also this here I have to put away because I don't want to have a mess. So the last two are now, two stains where I actually do know that they work quite well as well. One of them is called safranin. It's 1% aqueous. I bought the safranin powder from a microscopy shop, and then I added some distilled water. So, and it looks extremely red, almost like blood. Okay, so we add a drop here. Okay. So I'm going to label this S like safranin, and CV for crystal violet. Crystal violet is also a very strong stain. Okay, and which is blue. So in case maybe some of you have been following the live stream a little bit, a couple of months ago I not complained, but I simply mentioned it. Look at all those blue dots here on the surface. That's all crystal violet, which was very difficult again to remove. Look, this one has also lost its color, its stain already. Who knows which reactions are happening in here? So usually you also want to give it a minute or so. Okay, and let me prepare the other slide again. Okay. So just dropping a cover glass on here is not recommended. Everything is going to be so heavily pigmented and dark that you're not able to see any proper contrast. So washing it and destaining it is always important because once the stain has reacted, let's say with the cell wall or the cell's contents, it should not be washed out anyway. Right, so that's the purpose here. So let's maybe give it another minute or so. especially when the specimens have been cut so thin, then it really does not take a lot of time for the stain to diffuse into into the cells and into the cell wall. So I think this should be enough. So let's take this. I should have labeled it over here. I just realized, obviously. So it turns really red. Yeah, and as I mentioned, look, this one has retained its red color. I mean, honestly, I don't even need to label it because I know anyway that one of them is red, the other one's blue. And the other one here, let's pick this one here up as well. Look how incredibly pigmented it is. Some months ago, when I did some gram staining, I also used crystal violet and saffronin. Yeah? So those stains are also used a lot in bacteriology, in classical bacteriology to stain bacteria. So here we go. Cover glass goes on top of each one again. You know what? I think it might be easier to just add a little bit more water right now. So, and over here as well. Oh, you see, there's still a little bit of pigment stain coming out here. That's okay. And let's see if there's only one of them. Still not fully. This seems to be a fairly thick specimen as well. I don't know. We're gonna give it a try. Okay, it's a fairly thick specimen, so it looks pretty dark. But we're going to give it a try in any case. So let's try saffronin first. Always, of course, go to the scope view, microscope view. And this is how saffronin looks like. Let me turn up. Let's remove the... I think that's actually one of the more pretty, pretty ones. And we can now quite nicely... Okay, we ignore the air bubbles again. Let's go up here. Yeah, I probably should have destained it a little bit more. But let me have a look. No, I think I'm going to go down again with the magnification. I might retry this a little bit, maybe with a different cross section. Here. That's what I wanted to show you. So you see that over here again, we can see that the cell's contents have stained more red, and some cells seem to be more transparent, right? And this is those cells that have lost the contents, like those over here. That's actually quite nice, because what we can actually see again is the following: how the membrane has separated from the cell wall. So on the outside here, we see that the cell wall has now taken on a red pigment, and over here this seems to be maybe the cell membrane with the cell's contents, which is separated over here from the cell wall. So we're able to see actually like right through the cell. And the saffronin basically has also stained the cell's contents, and because most cells are actually red, it shows that actually most cells are actually still intact. So this is a little bit the thing. So let me quickly go down with the magnification. I might actually want to redo that with a thinner one. And this here is now crystal violet. Okay. You can also see that the crystal violet has now stained the cell's contents, but some of them appear to be way brighter, either because they were cut open or because the cell membrane has separated. So, but what I'm going to do is now I'm going to repeat this staining process, and where did I put it? And here it is. I'm going to repeat the staining process, and I'm now going to use thinner sections, because I would like to see if maybe I'm only able to stain the cell wall and not also the cell's contents. But if you compare it, for example, here, that's the outside. That's why this would be blue as well, had I not washed it in water. So let's do the following. Let's repeat this here. And let's have a look at how this actually works when I use a significantly thinner thinner specimen. So maybe I'm going to use one that was cut like a wedge, because there, where there is, it's going to be definitely a single cell layer. So I don't know. So maybe this one over here. So I'm going to put this in here. Okay, here we go. Yeah, same thing. So because it was now cut, how do you say, diagonally? So this means right at the edge, I expect the cells to be extremely thin. So this is simply as a reference. Look, maybe I have to turn this here also around. Okay, here we go. Now it's completely submerged, and then we repeat the whole process. We put again the whole thing on a different slide, and with a cover glass on top. So what I generally recommend is that's one of the nice things about hobby microscopy is to simply do a little bit of experimentation. Yeah, it's trial and error, and then sometimes things work out, sometimes things don't work out as you hoped. But that's the whole point. Even if things don't work out, like for example, certain stain not working, we've learned something, right? And for this reason, we basically appreciate that microscopy is of course also a hobby of learning, right? And even if things don't work, then we basically know that from now on it doesn't work, and we try a different stain. The only issue is now this one is already as a washing solution a little bit, I don't know. I might have to replace this, but no, you know what, I'm just gonna, the concentration is so low anyway that I think it might not make a huge difference. Okay. So, we still see that it's still red and still blue, even though over here this is a mix of all different stains. That's also something that you probably want to avoid, is mixing mixing everything. If it becomes too concentrated, then you probably want to exchange it, because then it might actually stain the specimen again. So we're going to do it with, again, a little bit of, we need to add a little bit more water here, otherwise the cover glass is not, see, there are lots of air bubbles in here. You know what, who cares? I'm only interested in actually seeing those few cells that have stained, and we're gonna simply use this now as a new reference. Then so where is the scope? When I say new reference, basically of how the cell wall should look like. And then let's hope for the best. I don't know. Where is it? Here we need to go down with the light intensity. So let's refocus. Here we go. Yes, that's actually what I wanted to show you. So that's the thing. Let's go down yet further and let's go up with the magnification. So that's actually the thing. Let's go all the way to the side here, and we do see that the background is a little bit red. Maybe this here is a bit better. Here we go. Yeah, so that is basically right at the where because I was cutting it diagonally all the way where it becomes very thin. You see over here that where it's so thin that most of the cells are actually cut open, and therefore they appear to be white on the inside. But the important thing is that you see that the cell wall definitely also now has a red color, and sometimes you still see, especially with the smaller cells here, you see that? This one here and this one here, they seem to be still intact more or less. So, and the others, the larger ones here, I've cut open, and therefore they've lost the cell's contents. And another thing that I just want to mention over here is especially those cells of the vascular bundle that we have over here. They all seem to be white on the inside as expected, because they are hollow tubes that carry the water. So for example, those cells here with a thick cell wall, I know that this is xylem, which transports water up the plant. And of course these are long tubes, and therefore these long tubes of course are hollow. So that's why the background is white here. Look, when you go up with the magnification, then you start to see the effect of the pigment yet a little bit more. So turn up the brightness. So so that's what do we learn from this? We learn from this that safranin does stain the cell walls. Again, if you want to experiment this, I bought safranin from a microscopy shop in the UK, in Great Britain. It's called Brunel Microscope. I always have to say it's not sponsored or anything, but that's actually one of my go-to places if I need microscopy stains and microscopy equipment and so on. Yeah, because many of the stains you simply cannot buy easily, or you can only buy them from specialized chemistry supply stores, but they sometimes do not sell to private people, only to universities and research institutes. So it's nice to have a shop that's willing to also send to private people. So it's called Brunel microscopes. The reason why when I focused, you see this? Yeah, it is because the specimen is so thick that it already touches the cover glass, the objective, and it kind of pushes down on the cover glass. That's why it kind of looks. Yeah. So now let's have a look at the crystal violet one. So I have to find it again. Where is it? Here we go. Here it is. Let's turn it up. So let's see if I'm also able to find the edge. There is basically, it's here. Here we go. Yes, here it's much thinner. It's too bright. Let's go down with the light. Okay. Yeah, so similar case. Look at this here. Over here, it's not stained. So evidently, maybe some of the stain either did not reach all of the parts. Yeah, that sometimes happens. Yeah. But also here, a similar case where the cell wall has taken on the stain. And now it's blue. Actually, this is actually a pretty nice one. Let's remove the arrow again. It's kind of disturbing. Yeah. And you can also see quite nicely how thick the cell wall actually is. This is actually almost an artistic. Take a picture of this and then put it up on your wall. So this shows again that look. The cells on the side, there is this abrupt, how do you say, so for whatever reason it did not take on the pigment, because there's such an abrupt, how do you say, change in color. Yeah, so what I'm going to do now is as follows. I'm going to just, concerning the staining, I'm just going to leave it at that. If you joined in a little bit later, I've just want to inform you that I've tried now 6 different stains, of which some of them worked quite nicely, others didn't work at all. Yeah, and that's simply something that I wanted to show you and also share with you, so that I can also learn something in the process. Yeah. Safranin and what we've got here, crystal violet, they are pretty strong stains. Yeah? And this is pretty cool. Look, it's actually very nice. Yeah, so I'm quite happy that this works. Yeah. So, and here again, interesting. Here again, that's very nice too. We can see again the contents of the cell. How the contents. I need to turn on the arrow again. How the contents of the cell here. Yeah. Has separated from the cell wall on the outside. And here we see that evidently different cell walls stain differently over here, down here in the corner, quite blue, and those over here not quite as much. Yeah. Here again the xylem stained very blue. So depending on the chemical composition of the cell wall, you have different degree of accepting or reacting with the pigment. So I'm looking for my words here. Okay, so I'm checking the time. So I've been doing this now 15 minutes. For the next couple of minutes, I would like to simply completely change the topic, and I would like to simply try to revive and rehydrate or regenerate my slime molds. This was actually also a request in my last week's live stream. The request was how are the slime molds doing? And I said, well, they're not doing anything at all because I have not revived them. So I would like to show you what I have been doing and what I'll be doing today. And if you don't know what slime molds are, or no guarantee that it actually also works, and we have to wait a few days to see if it actually did work. So I'm going to take this out here. I bought several months ago a slime mold, and it came on pieces of filter paper. And I've been growing them, and now it looks like this, and I need to explain this a little bit. Okay, this is filter paper. That's some cereal in the center, and you see those yellowish things. That's a dry slime mold. The slime mold will spread over the filter paper, and when you allow it to dry, then it looks like this. However, they should not be entirely dead. Now, I've not done this in a proper way. You have to keep it in darkness. The temperature has to be controlled. All of these things. I did not do that. I simply slowly let it dry, hoping that it's still able to survive. So that's actually one of the other things that I'm kind of trying to do is I'm trying to experiment on which conditions I'm able to get it back to life. So what I need to do therefore is the following. I'm going to take some scissors, and I'm going to simply take this here, okay, with the slime mold, and the rest here goes back into the, I've got a few more here. Here it is a little bit here as well. And this goes back in here again. And I'm going to now do two different experiments here. And I've got over here, this is just regular filter paper. This is, by the way, a coffee filter paper, right? If you want to make your own coffee. 2 layers, simply to give it more water. And you add a little bit of, I just realized I did not add enough water. Okay, here we go. So you will allow it to absorb the water, make sure it's flat, and we are already ready to go. So what I'm going to do next is I'm going to take this here. This is called the sclerotium. I'm just going to cut it in half, and I'm going to put it in here. It will absorb water, and over the next couple of hours, I'm going to see if it was able to get back to life, okay? And there will be a separate video on that. So that's basically number one, but there is also going to be a new thing. And no, I'm not going to be adding any cereal just yet. I've done this in the past. I've added some food already to the slime mold, but this caused the bacteria to grow too quickly. So you have to give it a couple of hours first for it to regenerate, if I'm able to regenerate it at all, okay, from here. So we're going to see. So number one. And number two, look at this. What is this here? Okay, this is an agar plate with some yeast extract, not yeast extract, actually I added some yeast. It's a homemade agar plate. And so there are plenty of nutrients in here already. The dots that you see on the surface here are not bacterial colonies. These are water droplets. And so this means there is already plenty of food in here. And I don't know if this is going to work. I never tried it. So I'll be adding this sclerotium directly to the agar plate here. Okay. And I know this is not entirely sterile what I'm doing, right? But that's enough. Okay. It's going to absorb the moisture, and I don't know if I'm able to revive the slime mold. That's basically something I would like to experiment. But you know what? Why not do the following just as a last little observation. Let's switch over to my stereo microscope. Where is it? Here it is. Let's have a look now up how this looks like under the stereo microscope. Yeah, it's the one. Yeah. And it's dry and it's now absorbing, and I hope that it's able to get back to life. And I'm hesitating now because I'm already seeing something that surprises me. This is actually indeed surprising. Do you actually see that it's already starting to run over here the edge? Could it be that it's already reviving that quickly? I don't know. I'm gonna monitor this, but honestly, this is what you're seeing is there was no time delay, so I did not interrupt the video. But look at this; it's already starting to. It has just absorbed the moisture, and it's now starting to appear to spread already. But this means that it actually has revived within, I don't know, a minute. Look. This is indeed strange. No, I mean, normally, because I remember when I got this, when I bought the paper and I did that, it took, I don't know, a couple of hours, yeah, for it to, yeah. But this one already started to almost to appear as if here as well. Okay, we'll see, and over here, and this is how I'm going to put the other one looks like. Okay, let's have a look. I don't know. This is here. It looks a little bit more cut off, and I don't know if there is not enough moisture yet. Moisture has not yet reached the sclerosium over here. You know what I'm gonna do is I'm just going to leave it as it is. I'm going to report back on this, and I'm going to, of course, try to grow a little bit more slime molds, and maybe we're going to do another slime mold episode in the near future. Yeah, I've been now talking for 1 hour. Normally after 1 hour I have this Q&A session, the question and answer session, which I'm not going to have today. What I'm going to do now is I'm going to say goodbye, but I would like to, of course, encourage you to leave comments, and yeah, the community is quite active and alive. So if you quickly want to add a couple of questions for me, and there's no possibility to add them to the live stream chat, then please use the comments section, and I'm quite sure that some of the community members have also responded to some of the questions here. Yeah, again, if you joined late, I have pre-recorded this live stream, and the reason is because of personal time problems. I was not able to do it at my regular time, so I pre-recorded this video here, yeah, I basically wanted to keep the weekly pattern. Yeah, I wanted to maintain the weekly pattern because people are joining in and they're kind of expecting this every Saturday, and I hope that this is also going to be fine. But I'll be present again live, yeah, hopefully next week or the week afterwards, and so on. If for whatever reason it doesn't seem to work out again for me, then I'll again upload a premiere, 1 hour long video, just like I've done today. Yeah. And I'm just going to quit talking right now. I want to thank you again that you joined me here. I wish you all the best. Happy microbe hunting as always, and see you around in the next video. Bye bye.